Looks like we got a couple viewers coming in. I think you guys can type comments in. So I'm gonna just put my name, and then you can find me mostly active on Twitter at Kevin Klatt. Oh, cool, we got a few people coming in. Can you guys hear me okay? Shy group. Can you guys hear me? Hello. I think we've got a couple minutes before we get started. If you guys have any comments or questions, feel free to type them into the box. If you can't hear me, tell me to speak up louder. I can. Awesome. Thanks, Tara. So if you guys have questions, feel free to type them into the comments box. I don't see too many yet. Or even just let me know what you want me to talk about. I'm happy to kind of just sit here and say anything about my career path thus far, but I think it'll be a lot more fun if you guys have some questions. Oh, Leah McGrath is here. Hey, Leah. Alrighty, well, it is 11 o'clock. So we've got about 30 people on right now. Cool. All right, well, I'll just start talking and maybe we'll get some questions going. Um, so I figured I would just kind of talk about my path to where I am now. And where I am now, spoiler, is I'm a postdoctoral associate at the Baylor College of Medicine in the Children's Nutrition Research Center. Um, and so I've done a good bit of work. I don't do any clinical work at the moment apart from some telehealth stuff on the side, but nothing inpatient. But I did my doctorate at Cornell University in the Division of Nutrition, and then I did my dietetic internship at the National Institutes of Health. Those are not marijuana plants in my window. I mean, you know how it's a financial struggle to become an RD, but it's not that bad yet. So so I actually did not do my undergraduate in nutrition. I did it in biological anthropology, and I didn't even really know when I was going to college that I wanted to get into nutrition. I had lost weight in high school and kind of got a little bit interested in it, but never thought about doing it quite academically. And then probably about my second year of undergrad, I realized that maybe I shouldn't be an international business major and wanted to switch over to the sciences and pursue nutrition in some way. But then my undergrad didn't have a nutrition major, and I didn't want to transfer and lose scholarships and all that kind of stuff. So I switched over to biological anthropology because it was the easiest way for me to get the sort of most of the DBD courses that I could possibly get done after I figured out what a DBD was, which took enough time as is, and did all my basic sciences coursework. And then I ended up going through that whole nightmare and did some of my DBD courses at Drexel University after I finished my undergraduate. And I taught there for a year as a TA and did a little bit of research, and then did some DBD courses, and then went on to Cornell, where I finished the last couple of DBD courses I had, and while I was doing my PhD. And then I finished up my doctorate, and as I was doing that, I applied for dietetic internships, and I applied to one spot, which was NIH. I ended up getting it. They have a very research-oriented dietetic internship. And then here I am. Now I finished up my dietetic internship at NIH last year, and now I'm in my first year of being a postdoc doing research. Does anybody have any questions about that so far? Apart from whether I grow marijuana. Anybody? Don't be shy. Alrighty. So I'll go ahead and just, oh, sure. Hey, Bridget. So you asked, can you talk more about the research aspect of the NIH dietetic internship? Sure. So research is kind of intertwined entirely into the NIH DI. So they have 4 students that they take a year in the internship, and you're in the clinical center, which every patient you're seeing there is pretty much on a research protocol. So it's sort of every single patient that you're seeing is a research subject, and we get a bunch of questions here. Cool. So you do have research components where you're doing your own little projects with sort of some existing data that has been collected, but a lot of the research that's done from within the nutrition department at NIH is more research support. So you are providing clinical support to the clinical research protocols going on that can be anything from very nutrition relevant things like non-alcoholic fatty liver disease to things you wouldn't typically think about, like a lot of the immunotherapies that are going on. You're just making sure that you kind of limit the noise in that data set by not allowing abject malnutrition to occur and doing your typical nutrition assessment. But then also we spent a good bit of time in the metabolic kitchen as well, where you're supporting other research studies that are going on within the intramural program at NIH, where you're providing food. in some sort of way. And that can be something very simple, like a low calcium diet for a study with a drug that needs to be on a low calcium diet, or it can be some of the work that Kevin Hall there does with the control feeding studies, looking at like how macronutrients influence energy expenditure. That can be, that is all supported by the research staff there that are largely dietitians, in addition to kitchen staff and things like that. So it was a good experience. It was definitely unique. There are not many metabolic kitchens left, and that's one of the sort of longest running ones. So you get definitely a different take on a dietetic internship at the NIH. I think I've passed Anthony Fauci, but I don't think I've had any really meaningful conversations or interactions with him, unfortunately. And then Morgan asked if I think there's choline deficiency has a role to play in intrahepatic cholestasis of pregnancy. That's an interesting question. It's definitely not something that has been studied very much. We don't have really much data on choline in pregnancy, which is why I had hoped to do it during or add to that literature base during my PhD. I had started a clinical trial of choline supplementation during pregnancy. But as far as specific recommendations about using choline to alleviate hepatic cholestasis during pregnancy, I've not seen any sort of major literature there about that. But choline is definitely important during pregnancy regardless. And so not something to be ignored, but I don't want to say anything about being specifically therapeutic because it's never been tested in a controlled trial, or I don't even think in an animal model of cholestasis during pregnancy. But it's certainly an interesting topic. Thanks for the questions. That makes these a lot easier. Okay, so why did I first get involved in Twitter, and what keeps me being there? It's like an existential question I ask myself quite often. I first got involved in Twitter actually because as I said, my undergrad's in biological anthropology, and during that's sort of the human population genetics, studying the evolution of the human species. And so while I was doing that and also kind of doing all the classes to head towards a degree in nutrition, the paleo diet was like super huge. And it was just sort of interesting to me, the disconnect between thinking about all the evolutionary factors that might have influenced humans over the past millennia, and then that there was a popular diet that was sort of just very simplistically thinking about what the forces of evolution were, and then evolution would have acted so that we would have one perfect diet for humans to prevent all chronic disease. Just doesn't really jive too much with our understanding of how evolution works. So I kind of got interested in the early blogging, and things that I did was about like some of the sort of misconceptions about how evolution works and how that interacts with diet. And then I begrudgingly joined Twitter because I had seen some other people who were using Twitter pretty regularly. Actually, I used to follow Roseanne Rust's blog and Colby Vorland's blog, kind of back when blogging was bigger than Twitter, and then got on and just kind of slowly used it as a platform and engaged with some other people who are pretty active, and ended up liking it and seeing that it was a place for a lot of information, but also misinformation. And there was an opportunity to kind of toss my voice in there a little bit. And I swear, I always joke with people, I learned more from Twitter than I learned in my graduate school classes, because there's just an incredible wealth of scientists and wealth of knowledge on Twitter. Really high level thinking can occur in different parts of Twitter. I'm not saying all of Twitter, obviously. I'll refrain from being political here. But there are definitely a strong scientific community who are doing a lot of really great science communication. And I think my motivations for staying on Twitter over time have definitely changed quite a bit. I've sort of gone through phases of being a little bit more interested in agriculture, and I was interested in genetic engineering and the kind of conversation around that, as that has waxed and waned over time. And I think more recently, I just stay on Twitter because I think I see a lot of people with thoughts and opinions on how nutrition research should be done. And as a nutrition researcher who's kind of bridged both doing cell and animal studies, but also doing human studies, I try and add a little bit of a voice there, and just sort of I'm hoping to move the conversation along in a way that's a little bit more productive, because it seems like nutrition can run in circles all the time and not really have an end goal in mind. Hey, Aaron, how's it going? I think got a few more questions. And Leah, does feel like I've known you forever. How do I boost my immune system? Oh gosh, I figured that immune boosting was going to be a topic at some point with coronavirus. Thank you. I'm going to avoid that as much as possible. Would I recommend any supplements like L-glutamine, beta-alanine, or citrulline malate to enhance athletic performance? I think there's a little bit of data for beta-alanine, but I kind of avoid sports performance kind of stuff. I guess my global take on a lot of the sports performance things is that they're often when you get into these really nuanced things like whether amino acids or citrulline are going to be beneficial. It's the sort of gains that you're getting from that are pretty marginal. That's something that maybe the Olympic athlete trying to shave a second or two off of their time is going to want to think about and value. But for the average person, it doesn't really enhance their athletic performance all that much. So I think for sports dietitians working with sort of athletes really trying to get the edge, following that literature really closely and understanding sort of the individual studies that might be relevant to the populations they're working with is something that they can do, but that's a little bit too granular from where I sit. I don't think that many ACSM really only highlight, so the American College of Sports Medicine really only highlights things like creatine, monohydrate that have some pretty good evidence as being a performance booster. But some of these things like glutamine and beta-alanine and citrulline malate, there's not a robust body of evidence to really have a strong opinion on it. So I don't recommend any supplements, and I also for athletic performance enhancement, and I also try to not work with athletes as much as possible, but that's just for my own personal preference, mostly because I personally swim and run, but I'm not really into bro science kind of stuff, and I'm happy to lift and just eat a little bit more protein, and that's kind of my goal. Sorry, that was probably not the most rewarding answer, but hey Karina, how's it going? I often mention that one food cannot fix everything. How would you design the ultimate nutrition trial focusing on immune tolerance with T regulatory cells as an outcome? Oh my goodness, T regulatory cells. I was actually just reading about those the other day, and how glutathione constrains serine metabolism in the T regs. I mean, Karina, I think probably my answer to that could be T-reg function. How do you design the ultimate trial to assess that? I don't even know if we have any good biomarkers of T-reg function. I mean, ideally with any nutrition trial, you would want some sort of dose response design with well-defined comparator groups. So I guess we would have to come up with some sort of hypothesis about a nutrient that we think relative to another nutrient. It's easier to work with micronutrients because you don't have the calorie issue. So when you have if you wanted to say saturated fats influence T-regs function, you'd have to do a really complex trial comparing saturates to monounsaturates, different types of polyunsaturates to really get some sort of idea about whether it's specific to an individual nutrient, whether that nutrient uniquely negatively influences the T-reg cell function, or if it's just only compared to that other nutrient. So I'm really trying to think of immune health, but lacking a biomarker other than T-regs. I mean, I think you can always sort T-reg cells from, that you can obtain from the body, and then do some ex vivo kind of work with them where you look at their function, or even just kind of isolate them and then maybe do some RNA sequencing, or put them into culture and then look to see what sort of cytokines they produce after doing some sort of a well-designed diet trial. So T-regs sort of constrain, I guess, immune function that is, their job is to keep the brakes on the immune system a little bit and make sure other types of T cells don't become overly inflammatory. And they're implicated in a lot of autoimmune diseases, like especially in the gut, where you want Treg function to be normal so you don't get like inflammatory bowel disease and things like that. That's a super like dumbed down version of what Tregs do. They're pretty complex, but in general, we think of them as maintaining a healthy immune function and not letting the immune system get out of control. So yeah, I would do some sort of trial. That would probably be a pretty complex feeding trial, and then just profile what the Treg cells are doing, what cell surface markers they're expressing, maybe put them in culture and see what cytokines they produce. But at the end of the day, I think you also want some sort of functional outcome in that patient population that you're looking at that's directly related to that we know that Tregs are involved with, because that's at the end of the day what you care about the most is those patient outcomes. Whether it's truly Treg mediated or not is really hard to do outside of an animal model. So in animal models, I mean, you could maybe do something more like this, where you isolate different T cells after some sort of immune or some sort of dietary intervention, and then infuse them into maybe like a rag knockout mouse that's immunodeficient to see how diet influenced T cell function. But there are so many ways we could go about doing this and so little funding. I figured you were trolling me a little bit, Karina. All right, let's see who else we've got going here. These comments, I think, come up a little bit slowly. Probably just bored everybody talking about Treg cell function. I think we lost like 10 viewers. Anybody else have any questions for me? I can talk about some other stuff that we're doing right now too. So I've got a couple trials that I'm wrapping up at the moment. What is next for me? Oh gosh, that's an interesting question at a time of the coronavirus and being unsure what's happening with funding. I think I'm staying at Baylor College of Medicine for a little bit, although my current advisor may be moving. So stay tuned there. A lot of what I'm interested in generally now is looking at how we can kind of bridge some of this basic science thinking and preclinical approaches in a smart way and translate them to inform new diet trials in humans. So some of the work I did in my dissertation was looking at existing known nuclear receptor ligands, that sort of ligands bind the nuclear receptors and activate them and make them more, depending on the nuclear receptor, it does a whole bunch of different things, but seeing how diet influences the production of those ligands, and then thinking about whether diet could be therapeutic or potentially detrimental in different contexts by their influence on nuclear receptors. And certain nuclear receptors have very clear roles in disease states. So like one of the ones that we study is LRH1, which regulates local glucocorticoid synthesis in the gut and has anti-inflammatory action, that there's a lot of interest in agonizing that nuclear receptor and making it more active in inflammatory bowel diseases. So one of the things that we're interested in now is whether you can either develop both drug ligands to make that nuclear receptor more active, or use diet in some way to make it more active and have a targeted therapy for inflammatory bowel disease. Because right now, I think most people are probably familiar that there's exclusive enteral therapy that seems to be somewhat efficacious for inflammatory bowel disease patients. It's much more utilized in Europe than it is in America. It's hard to get insurance coverage for it. But we are looking at, in preclinical models and some in mouse models, what might be some nutrients that agonize this nuclear receptor, and then will it have any benefit in treatment of patients potentially. So trying to think of things that could translate into clinic pretty easily. and have sort of value for patients. Because right now, I think with inflammatory bowel disease, people who are familiar with functional GI disorders and that whole world in general, and IBD, there's not a lot that we know about. We know so much about things being important in the gut, but when it comes down to how do you direct people's diets to actually improve outcomes, there's not all that much that we can offer. And even with things like exclusive enteral therapy, it's sort of formula independent. So there's not a specific component of the diet that we've isolated to say like eating more or less of this would be therapeutic. Somebody asked, any thoughts on anti-nutrients and PEDs like phytates and tannins? That's a really kind of hot topic in the literature, and it's been for a while, but sort of the idea that phytates and tannins can reduce the absorption of minerals. What's sort of surprising in that literature base is how much it's all based off of single meal studies. So, or very short-term studies showing that eating phytates or eating tannin-rich foods can block the absorption of iron. But there's been some controversy in the literature that's not really panned out as to whether the long-term consumption of those dietary components or foods rich in them, like whole grains, actually negatively impacts mineral status, and iron being the one that people typically are most concerned about. So I think the sort of consensus that's not based off of extremely robust data is that, if you already have consistent low iron intake, phytates and tannins are maybe going to exacerbate your iron status, but they alone aren't the cause of iron deficiency anemia. They just kind of worsen it. But again, those are based off of single meal studies. And there's been one or two studies, not really in the developing world context, that suggest that whole grains really don't have a negative impact on iron status and might be actually beneficial. So I think this is something that all of nutrition science has to learn from, is that shorter term studies, we kind of underestimate the body's ability to adapt, or are not able to predict what the body does to adapt. And we should be just very tempered in our conclusions, because there are tons of studies with really complex stable isotopes showing that like the phytate and tannin content of the diet can inhibit iron absorption in a single meal. But we don't have many long-term, like 12-week feeding study interventions with different levels of phytates and tannins showing that it detrimentally impairs iron status in the long term. So for kids, I guess you asked specifically in PEDS, in the developing world context, I think we're much more concerned about them. In the context of a generally well-balanced diet, there's not a ton of reason to be concerned. And I think this is something too that you can use a little bit of common sense, like maybe don't take your iron supplements with high tannin foods like coffee, and space those things out, because it really seems to be their presence in the GI tract at the same time that matters the most. So if it does seem to be an issue, or you're suspected to be an issue with a patient, trying to have them separate phytate-rich foods with mineral-rich foods can be one strategy that you try out and see if there's much of an improvement, as long as it's not super burdensome for the participant. But relative to things like inflammation that have a very large effect on non-heme iron absorption, I think phytates and tannins are relatively small factors. If you had a magic wand and could choose how research funding within nutrition were spent, what changes would you like to see, or what would I like to emphasize? I sort of agree with folks like Kevin Hall, even though it doesn't sound very sexy, that we really need a large commitment to well-done controlled feeding studies that can be done for a long time. Controlled feeding studies are kind of our gold standard, and in a perfect world, we'd like to do them for years and years and years, and that's not going to be possible. But a lot of the literature, even for the effects of fats on lipoproteins and things, most of the studies are like 2 weeks long or a week long. There's only a few that go much longer, and they're often not super well controlled. So I think I would love to see the government kind of invest in large scale facilities that people can live in for long periods of time, and everything about their food be controlled. And not only would that help us have more quality controlled feeding data, but we can develop better biomarkers for our free living population studies and go from there. And you can test everything from foods and nutrients, supplements, all in that kind of context. But you really need a commitment to doing very well performed diet and feeding studies well in some sort of controlled setting. And we just don't really have that. We used to have that with the generalized clinical research centers that have largely been defunded. So you have a few small controlled feeding studies that happen here and there, but just you look at the field of medicine and what they're able to do with very, very, very large controlled trials. And obviously it's easier for them with pills, but we need some sort of larger commitment to controlled feeding where we know what people are eating at the end of the day and can look at biomarkers. Will I share my secret to staying up to date on so much of the literature at once? Yeah, Twitter. And honestly, I think it takes a lot of putting in the work early on in your training, and it gets a lot easier over time. The more you kind of increase your breadth of knowledge, the more it's easier to just update kind of what you were thinking. So I would say go broad, particularly for those of you who are in training, not taking a knock at any particular nutrition textbook, but if you compare nutrition textbooks to medical school textbooks or other medical physiology textbooks, usually the nutrition pathophysiology section is like a little blurb, a little quickie that really undersells what's happening in the disease process. And I think it makes it's much more enriching if you have the time and the energy to look at, what a first-year medical student is learning about a disease process. And that for me was what I did, and it helped me to I just found even like a not the most recent version, but an earlier version of an internal medicine textbook, and read those pathophysiology sections, then came back to the nutrition textbooks to figure out what the MNT was. But it was quite helpful for me to kind of fuse medical and nutrition textbooks for sort of really establishing a larger base of understanding of the human body and different diseases and things like that. And then your dietetics training is perfect for understanding food and nutrients and how they might fit into those diseases. We could have used the COVID quarantine for a huge controlled trial. That is correct. Let's see, any other questions here? And also, follow smart people on Twitter. It's always good to keep in mind that there are smarter people than you out there. And I think the way I approached Twitter was to follow a few people in one different area. And then when it's like kind of got a handle on that, I'd follow some other people in some other areas and just slowly build up over time. I think it's overwhelming to dive into the entire literature base all at once and try and, feel like you understand much of anything. But a huge part of it is just regular exposure to seeing these words and things over time, and not being afraid to ask questions of people and questions that you feel like you should know. Any resources I want to give a shout out for, like websites? So obviously the Build Up Dietitians page shares a lot of good resources. There were a lot of, blogging used to be really big, and I feel like I learned a lot from blogs over the time. Everything from following people I really agree with to people I don't agree with, just to challenge my sort of critical thinking. I remember the Food and Farm Discussion Lab group on Facebook it was interesting to see some of the nutrition topics there, and they also talk about agriculture and things. So if you're interested in food, the Food and Farm Discussion Lab page is really great. Medical school textbooks that you can get, things like the principles of internal medicine and things that are just the basics of what an internal medicine physician is expected to know, are reading whatever area you're working in or whatever area you want to work in, kind of reading what the medical doctors were taught, I think is always super important, because at the end of the day, you're going to have to talk to medical doctors. Medical doctors really like to talk to people who are speaking on their level. And I can't emphasize that enough, that you kind of need to, it's always good to come off smart. So take the time, read what the medical students were taught, know their language, and then communicate nutrition in that language to medical doctors. So that helped me a lot in clinical, I think, and getting a lot of respect from the medical team, just to be able to speak their language and know all the things that they're doing and the expected course of treatment, and then knowing where nutrition fits in alongside all those other things. I think that's what's hard about nutrition, is we have to know what everybody else is doing, and then we have to know where we fit into that context. And a lot of our schooling only teaches what the nutrition is supposed to do, but it doesn't necessarily teach what it doesn't tell you what the medical people are doing, and knowing where you fit into the medical hierarchy, and where you fit in with that specific patient, where how well nutrition is going to be prioritized is super important. What is my perspective on balancing science and research with faculty responsibilities and working within higher ed? Oh gosh. Well, I'm not a faculty member, and I don't think I plan on becoming one. So that's sort of the easier part. I do not envy people who are planning on going a typical professor route right now, because the expectations for you to teach classes and run labs and have an outside life and be publishing all the time and be getting grants and training students and being an advisor. I don't know who has that kind of time, but I think there's a lot of people considering alternative career paths, because the typical professor position, people are just getting sort of overloaded. At the end of the day, we're trained to be scientists, and I think the faculty responsibilities and things are all you kind of have to learn on the fly, and you can't be really expected to be good at it, but I think a lot of people are. It's not to discourage anybody from going that route, but I think it's just a reality that it ends up being a humongous sacrifice, and you have to have a family situation and just a life situation and a mental health situation to be able to tackle all those different things that you're expected to do. And it's a huge sacrifice at the get-go. So I'm glad people like you out there, Dr. Hobb, are tackling all that. I wish I had some sort of secret, but I don't think the tenure track of life is for me. I'm kind of trying to figure out ways to just keep doing research and have that be my primary role, while also having a salary. So anybody else here? I know these things are typically a half hour, so I don't want to take up too many people's time, but I'm happy to stick around for a bit. See, there was this talk of a 2005 Ashworth and Golden paper on moderate malnutrition. It's work in underprivileged communities. Our main foods are advocated our highest maize meal. Yeah, I think that's back to the phytates conversation we were having before. And I think a monotonous diet, it tends to be a major contributor to malnutrition. And people point to sort of tannins and phytates maybe exacerbating that sort of situation. But I think fixing the monotonous diet is really the biggest priority there. That's right. Everybody wash your hands. Everybody thank your local healthcare workers who are going in and facing this virus. They are really health heroes right now. Cool. Well, you can find me on Twitter at Casey Klatt. I put it as the pinned comment. It's pretty much the easiest way to reach me. I don't check my other DMs every single day if I don't follow you, but I check them pretty regularly, so I can hopefully answer any questions you guys have. And you can also catch me by email. I guess I can type that in. It's just my last name at bcm.edu, which is klatt@bcm.edu. If you want to email me any questions, if you want to collaborate on anything, I like to study all sorts of things. I like to stay interesting. So I'm also happy to just kind of talk about research topics that you might want to throw around. I'm pretty passionate about dietitians getting more involved in research, and I know it can be a little bit scary at first, but anything we can do to kind of lower that bar and really elevate the role of dietitians in research is a big passion of mine. So happy to help out as much as I can there. Alrighty. Thank you all for coming and for engaging. This was fun. I'll have to do it again sometime. All right, all. Have a good day. Stay safe. Stay socially isolated, well, physically distanced, but check out for your own mental health during this time too. Have a good rest of your Friday.